good morning to everyone. I would like to welcome you to our session on immune system modulating drugs. Of course, there will be some presentations on histamine, glucocorticoids, the old folks. But of course, because inflammation, as we all know, is a protective response of the body, what we will try to see is whether this can be regulated or what information we have to date about this regulation of the inflammatory response with regard to immunomodulating agents. So I think we can start the session.
Sure.
Okay.
So I'll introduce our first speaker. Professor Katerina Tiligada received her bachelor's in University College Cardiff and a PhD in University of Wales College of Medicine. She then joined the University of Athens in Greece and established the Immunopharmacology Unit of Inflammation. She will be sharing the role of histamines and antihistamines in the immune regulation.
Thank you very much. I must tell you before starting that this was actually a session that was proposed by Francesca Levi-Schaffer, who was the chair of ImmuFar. At the time, there was the ImmuFar section of IUFAR, and I was co-chairing the section. And we would like to thank the organizers for accepting our proposals. So in my talk, what I'm going to do is present some evidence about histamine and antihistamines in the immune system. And of course, I will start with what everybody knows, is that histamine is released from muscles to mediate allergic reactions via binding to H1 receptor. I think that's the dogma that everybody knows. However, all these years that people research histamine, and we have people in this audience that have worked on this field, we just now know that histamine is a developmental, highly conserved endogenous biogenic amine. It's distributed all over the body. It is produced by a wide range of cells and exerts diverse actions via binding to 4 receptor types that are involved in multiple responses. Now, if I can change the slide. Now, these are the 4 receptors. And of course, they are distributed in almost every cell of the body. And we know that at least H1, H2, and H4 receptor, they are found in immune cells, particularly dendritic cells. We do not have very good evidence about the H3 receptor, although we know that some of the immune cells have this receptor on their surface. And of course, the H4 receptor, which is the latest that was discovered, is mainly distributed in immune cells. Now, what we know is that the H1 and the H2 receptors are what we call low affinity receptors for histamine, while the H3 and the H4 receptor, they are the high affinity receptors. We need lower concentrations of histamine to activate them. And of course, if I may just say, just very roughly, the H1 receptor is the one that is involved in allergies. The H2 receptor mainly is involved in gastric acid secretion regulation, but you will see that it appears to be involved in immune cell differentiation as well. The H3 receptor is the main autoregulatory receptor, autoreceptor in the CNS. And the H4 receptor is what we like to call the immunomodulatory histamine receptor. And of course, this is a mast cell, and the mast cell can be activated by various triggers, amongst them via the Fc epsilon receptor. It can be activated by antigens or allergens, but of course it can be activated by other triggers. Now, once the mast cell is activated, there is a plethora of mediators that can be released, and of course histamine is the main mediator that is released, and what it does by acting through the H1 receptor, it affects vital processes of the body like vasodilatation and vascular permeability, smooth muscle contraction, and of course it mediates the will and fear reaction and anaphylaxis. Now, I must tell you that, or rather remind you that mast cells and basophils are the main professional histamine synthesizing cell, but as I said before, there are a lot of cells that really produce histamine. And of course, this is a graph that shows that in hypersensitivity reactions, we know that histamine is released within the first 30 minutes, 20 to 30 minutes, after triggering mast cell degranulation or after contact with the allergen. Yet, as it is shown here in patients, I will show you a lot of real-world evidence that we have been working on all these years. So these are patients that report hypersensitivity reactions to anti-inflammatory drugs. And these are the basal levels. That means that they do not have the reaction at the time of measuring their histamine levels. And of course, what we saw, although the range is quite wide, we saw that these people have increased levels of basal histamine in their blood. And of course, this is another study that what we tried to do by using this excellent model, human model, of studying hypersensitivity reaction, and it's beekeepers. Beekeepers are normally healthy people who might have hypersensitivity reactions, type 1 hypersensitivity reactions to bee sting. And what we really saw is that there was a positive correlation between serum histamine and IgG4 levels, and this supported the contribution or the implication of histamine in the tolerance environment. I must remind you that class switching towards IgG4 is mostly associated with Th2 responses. And of course, we saw that higher serum histamine and IgG4 levels were seen in tolerant individuals compared to non-tolerant beekeepers. So these are some indications that histamine is not only involved in type 1 hypersensitivity reactions, but it might regulate some immune or inflammatory responses in the body. So these are some examples, and if this is a dendritic cell, we know that dendritic cells express H1, H2, and H4 receptors. There is some evidence that H3 receptors are also expressed on dendritic cells, and of course, histamine in neurons is involved in this regulation in the periphery, in the peripheral nervous system. But I'm not going to go into that now. So it seems that by activating H1 and H2 receptors, we get this balance between the T cell subtypes and subclasses. And apparently, what we know now is that histamine, by acting on H1, H2, and H4 receptors, it modulates T cell differentiation and chemotaxis. And in particular, in the skin, histamine is very high in the skin, as it is in the lungs and in some other tissues, of course, in the gut. So dendritic cells, they, by acting, histamine acts on the H4 receptor, and it seems that it promotes chemotaxis of monocytes. And of course, CCL2 is a chemokine that, a cytokine, a chemokine that is actually released and differentiates monocytes to macrophages, and it seems that by acting, histamine by acting or orchestrating the effect of these cells, what it does, it helps the transition from acute to chronic inflammation. There is some evidence on that, but it remains elusive, this is the take-home message from this slide, whether histamine receptor expression in dendritic cells is altered during maturation, or if various subsets of dendritic cells actually up or down regulate distinct histamine receptor under specific microenvironmental influences. And my next slide has to do with mast cells. We know that mast cells express histamine receptors, H1, H2, and H4, and in particular, H4 receptor is highly expressed on mast cells. And what we think is that this receptor actually autoregulates the amount of histamine that is produced and released by mast cells in an autocrine manner. And of course, this H4 receptor, when it is activated, it induces mast cell chemotaxis. And what happens is it recruits more mast cells into the inflammatory site, and leading to more and more chemotaxis through all histamine receptor orchestration, and then it again leads to chronic inflammation. Now, of course, eosinophils are very important cells in Th2 type inflammation. And here again, we are not sure exactly what histamine does. What we know is that, again, H1, H2, and H4 receptors are all expressed on eosinophils. But what is very important is that the inhibition of chemotaxis of eosinophils seems to be regulated by the H2 receptor when histamine concentration in the microenvironment is high, like the lungs, and it is enhanced via the H1 receptor when histamine concentration is very low. So we can conclude, for the moment, that there is a dose-dependent histamine-mediated eosinophil chemotaxis through orchestration of these three types of histamine receptors. And the more important part is that there is H4-mediated production of IL-31 in human eosinophils maybe is implicated in pruritic effects that we see in skin diseases. And of course, the take-home message again from this slide is that there is controversial data on histamine-mediated eosinophil activated. And now I come to another type of cell, which is the human monocyte-derived M2 macrophages that are mainly involved in some skin diseases. Again, you see that they express H1, H2, and H4 receptors. Now, what are the data we have so far? CCL18 is one of the molecules that is produced and is found in high concentration in skin diseases like atopic dermatitis. And what is shown in the literature is that histamine, via action on the H2 receptor, it increases, there it is, it increases Th2 cytokine-induced CCL18 upregulation. It is already upregulated by Th2 cytokines, but then histamine further upregulates it. And by using various histamine receptor antagonists like the H1 antagonist 2-pyridylethylamine, or the H2 antagonist like anthramine, or the H2/4 agonist 4-methylhistamine, or these new compounds that Holger Stark in Germany has produced, the ST 1006, we can see that this is a selective effect of the H2 receptor and not the H1 or the H4 receptor. On the other hand, we see that histamine downregulates via action on the H4 receptor C3A. This is a molecule that, I remind you, can act on mast cells and induce the granulation. But in this case, histamine downregulates regulates this molecule in these differentiated macrophages. And moreover, histamine, by acting again on H2 receptor, it downregulates the receptor for the intracellular concentration of the receptor for IgE. And as you know, rats or rodents, they usually have this receptor on mast cells, but in humans, it can be found in other cells as well. So we really do not really know whether histamine has anti-inflammatory and or pro-inflammatory functions in skin diseases, but so far the evidence supports the hypothesis that histamine regulates the function of these cells with regard to disease severity. So that's another very important function that we have to have in mind. And of course, in another disease, skin disease, in chronic urticaria, these were, the previous slide showed some ex vivo or in preclinical results, but this is real-world evidence. This is patients with refractory chronic urticaria, and what we saw is that in serum, histamine levels do not change compared to control subjects. But when we measured histamine in the whole blood, including the cells, then we saw that in the non-responsive patients, the ones who actually get antihistamine, anti-H1 antihistamine treatment, they actually have higher levels of histamine in their blood compared to the ones that are responsive to therapy. And when we try to see why, we just saw that this was correlated with the peripheral blood monocytes, which, as I said before, when they differentiate to macrophages in the skin. And this was a very nice work that we did together with Markus Maurer in Berlin and Paul Chazot in the UK, and we saw that actually there was a variation in the epidermal and dermal anti-H4 receptor distribution in responders versus non-responders. So this is the last piece of real-world evidence, because when we got patients with psoriasis, which is another disease that affects the skin as well, we saw that normal subjects had higher histamine levels than psoriatic subjects. And when we analyzed, this is a very preliminary work that we did, we analyzed it, we saw that as the disease severity was more important, then we had less and less histamine in their blood. And when we looked at the basophil count, to our surprise, we saw that people who had psoriasis had a lot more basophils than the ones who didn't, than the normal controls. So this wouldn't correlate, but we saw an inverse correlation between the amount of histamine in the blood and the number of basophils in the blood. And of course, we looked at the literature and we realized that in Th2 responses, there is evidence that basophils, they don't release histamine, but they may release Th2 cytokines instead. So this is a nice starting point to see what happens with histamine and basophils in skin diseases. Of course, after having presented these very selected points to illustrate the potential of histamine and histamine receptors to modulate the immune and inflammatory responses, I will just present some of the clinical data. And of course, preclinical studies and real-world evidence were very encouraging and led to the development of histamine receptor targeting drugs, sometimes with bispecific, with specificity for H4 and H1 receptor or H4 and H2 receptor. And everybody was really very happy to advance these molecules to clinical trials. However, until now, clinical investigation outcomes failed to translate preclinical findings into therapeutic benefits, although many clinical trials have been run with antihistamines. The only one that is currently active is a phase 2 trial to evaluate the efficacy and safety of an H4 receptor antagonist developed by Leo for the treatment of adults with moderate to severe atopic dermatitis. Of course, there is another clinical, it's a retrospective trial that is active at the moment and has to do with the use or the effect of H2 receptor antagonist in COVID. But I'm not going to discuss. We can, if anyone wants further information, I'm going to talk about it. So there are some key issues that really limit the translational potential of this preclinical and real-world evidence, preclinical research. And first of all, we need to know what is the endotype-related implication of histamine in all these controversial data that I have selected to show today. And of course, there is the differential control of histamine receptor expression in a given cell type at a given local microenvironment. And because we are pharmacologists, we know that histamine receptor pharmacology, like constitutive activity, and histamine receptor ligand properties, like biased signaling, may have affected the outcome of these experiments. And I would like to stress that the prototype H4 receptor selective antagonist J&J 77-120 has been used to characterize all the physiological role and the pharmacological properties of the H4 receptor when it was discovered in 2000. But then we couldn't understand some of the discrepancies that we could see in the literature. And between us, we had a cost action that Francesca and I really participated in this, and that was a very vivid and very well-organized effort to characterize the receptor, but most of us used the J&J compound. And then after a while, we realized that J&J is biased towards beta-arrestin pathway and exhibits partial agonistic activity, whereas when it acts on G proteins, it has a partial inverse agonist activity. And of course, it has a partial agonist activity on the rat and mouse receptor. So we have to be very cautious when we interpret the outcome of in vivo J&J compound experiments, that there are a lot in the literature. And just to give you some evidence on this, you see that these are rats, and you can see this differential effect of J&J compound in systemic inflammation that was induced by CFA, by completes Freud's adjuvant. These are Wistar rats, and you see that these are the normal histamine levels in various tissues, and when you give a J&J compound, you get increases in these tissues, similarly to the CFA, to the effect that we see after systemic inflammation. And although in some of the tissues we see no difference when we treat the rats with a J&J compound, then in the conjunctiva, we see an effect, a statistically significant effect of reducing these histamine levels after giving the compound. And finally, another very important consideration is that we have to consider strain, species, sex, and tissue-related differences in histamine levels and responses. And this is really a comparison between two strains. This is Wistar and Sprague-Dawley, male and female rats. And you can see that the levels of histamine in various tissues is not the same in both strains. And of course, here are male rats and here are female rats. And the first column, this is the control. They haven't been treated, or they treated with a vehicle. And the next column is the column that shows the histamine levels in rats. This is Sprague-Dawley rats that have been treated with LPS, and we have systemic inflammation. And you see, for instance, in the hypothalamus, the response is completely different between male and female rats. And the same is true for cartilage, whereas in the conjunctiva, we have the same response. So depending on the setup, the experimental setup, we get different conclusions of what the histamine receptors do. And of course, I have a lot of evidence to support that. I have just selected another two points. One is this excellent work by Gabriella Corucci in Parma, who actually took all the time to analyze what happens with a J&J compound in various mice, and you see that when she induced ear edema in mice, you see here that in CD1 mice, you get a reduction in the edema, whereas in bulb C or in C57 or in NMRI mice, you get no effect. So depending on the experimental model, then you get different effects of the compound. And moreover, finally, here is the J&J compound given to bulb C female mice and Perros. And it seems that the J&J compound, the H4 receptor ligand, I'm not calling it agonist or antagonist, as you realize, reduced the Th2 responses in induced airway inflammation. But then again, when the same compound, the agonist for the H4 receptor was given to the same model, we don't know here from the publication whether this was female or male, but we saw that it increased the upregulated Treg. So actually, we get different results for the H4 receptor, depending whether we use the J&J compound or 4MH. So there are, of course, other factors that can influence this effects of histamine, like the poor experimental models and biased or misinterpretation of findings. This is a work that we are doing in the European Histamine Research Society, and try to understand why we see all these discrepancies and contradictory results. Of course, We all know that there is a great difficulty in publishing negative data, and there is a lot of clinical outcomes that have not been disclosed so far. Of course, fragmented biomedical research and lack of collaborative efforts and failure to integrate evidence and data from all these studies and being more analytical rather than synthetic in research, there are some of the factors that we are not in a position at the moment to say what histamine does in immune regulation. So these are the conclusions: that histamine is a potent endogenous amine that is involved in homeostasis. It exerts multiple opposing or complementary effects on immune, muscular, neuronal, epithelial, and endocrine cells. There is a lot of evidence that supports the implication of histamine in a dynamic regulation of immune responses by acting on distinct histamine receptors that are expressed on immune-competent cells in a local microenvironment. And what I would give as an example is that histamine influences the Th1, Th2 balance and polarization, and influences the differentiation and or the recruitment of effector cells in immune-related inflammation. Now, considering this wide distribution and the many actions of histamine, as well as the wide yet differential cellular distribution of histamine receptors, we need to standardize the experimental models, to defragment and integrate the existing data, and to evaluate the translatable preclinical findings that can be exploited in the clinical development for the benefit, of course, of the patients. And this is actually, I would like to thank very much all these people who have worked the last 30 years in my lab on histamine, and of course, the members of the European Histamine Research Society that lead immunopharmacological research and related drug development and clinical evaluation. I would also like to thank the British Pharmacological Society and the University of Athens, who sponsored my trip to be here, and of course the organizers for giving us the chance to present this talk. And finally, I would like to thank you very much for your attention.
Hi, so we open the floor for questions for Professor Tiligada. Sorry, yes, we can hear you. But could you please introduce yourself?
So, Raymond Kambata from Queen Mary, University of London. Basically, I had a question about anti-histamines, so H1 antagonists. That's used a lot, obviously, you can buy them over the counter. And I wondered if, I would assume that you get raised plasma levels following taking those. So would that then be hitting H2, H4 receptors? And do you see any increase in incidence of things like atopic dermatitis or any kind of systemic diseases?
Well, you know, all the H1 receptor antagonists or ligands, they have been reevaluated regarding their biased signaling and their possibly partial effects on the receptors after the discovery of the H4 receptor biased, beta-arrestin bias. So most of them, they don't seem to be antagonists. They are partial they are inverse agonists, really. But what we have seen is in various studies that we use as controls, people who actually get H1 receptor antagonists, as we know, antihistamines, we don't see any difference in the peripheral blood levels of histamine. But I must stress that histamine is a tissue-acting hormone or amine. So, and as we all know, the blood is the homeostatic tissue of the body. So we cannot, as you understand, get a lot of samples from patients that have got this. As you saw, however, in people who are responding and non-responding in chronic urticaria, then we have differences in their basal levels, depending on whether they react to H1 antagonists or not. But we don't know, because all these are patients who have already got their therapies, their treatment. We don't know if this is an effect of the H1 antagonists or that's the basal levels. But so far, there is no evidence that blood levels, histamine levels, are changed after the administration of H1 antihistamine. And I have to say in this part that there is a lot of literature that is not to be trusted because it depends on the method that one measures histamine. Histamine is not an easy amine to measure. So there is a lot in the literature, particularly clinical data, that I'm not so sure if they can be trusted.
Thank you.
Hi there, Grant Drummond from La Trobe University in Australia. I'm really sorry if I missed this at the start of your talk, but you spoke about histamine and T-cell polarisation. Do B cells express histamine receptors, and does that influence their antibody production or cytokine production or anything?
Well, at least the H4 receptor is expressed on B cells. Now, as you know, I mean, T cells are more researched than B cells these days. What we know is that there is some evidence presented in the HRS that what the H4 receptor does, it may be implicated in the class switch, particularly in IgG4, not IgG1. So, but there is not much information on that.
Hi. Thank you, brilliant talk. I'm Daniela from University of Glasgow, and my question is about, do you think that the difference in male and female mice or rats can be related to the hormones?
Yes, there is I didn't have time to go through everything, but there is a very nice publication more than 20 years ago, or roughly 20 years ago, and one has really investigated the axis, actually the stress response axis, not the sex hormones, with mast cells and histamine. And it seems that at least in this axis, there is a lot of regulation of the immune response that is related to CRH. But we have done a lot of work on rats mainly, not on humans, with regard to the changes or the differences between male and female rats from the day they are born towards adulthood. Now, to be honest, according to our results, I don't see any difference, and I understand that because this is a very vital regulatory molecule, at least developmentally, then I haven't seen any difference between the two sexes, or genders, or whatever they're called these days. Yeah.
Thank you for a great talk. My name is Blaise Bilek, and I'm from St. John's University. My lab's recently started working with a croton oil model of ear inflammation. And I'm curious if we wanted to know the receptor subtypes for histamine within the tissue. Could we do immunohistochemistry for that, or is it better to do radioligand binding? Can you comment?
Well, there are some problems with the antibodies, as you know. And we have done a lot of work with the ADH4 antibody, and it's not consistent. So what we really try to do is, we do immunohistochemistry because everybody starts with this point, but at the end, what we try to do is do a lot of pharmacological experiments with agonists and antagonists, now that we have more information about the pharmacology of these ligands, just to verify or not. But it's very important, as you saw, which model you're using.
Definitely, thank you.
Yeah. I think that's it.
I guess I have one last question for myself.
Okay.
What were the effects of antihistamines in asthma?
Well, as we all know, antihistamines do not work in asthma. Okay, and although there is plenty of histamine in the lung.
Yep.
And of course, all the clinical trials with the H4 receptor antagonists in asthmatics, they just failed, not only in efficacy, but I must tell you that there are a couple of clinical trials that failed because of safety, because these compounds, they induced granulocytes, and there was a problem. Now, what we think is that particularly in asthma models, because the results are very controversial, we really do not know what the orchestration of histamine is, and as you know, very well, it has to do with the microenvironment. For instance, if this is an eosinophilic or a neutrophilic or a non-eosinophilic or neutrophilic asthma, then we have different results. But more importantly, we do not really have a real model to test that is not induced asthma. We don't have rodents that, well, there is a strain that can have spontaneous asthma, but this is a very difficult situation to judge. That's why we cannot translate these results into humans. But so far, all compounds failed.
Cool, thank you. So let's thank Professor Tiligada for her talk.
Thank you. Can you hear me? We can go on with Carlo Riccardi. Carlo is a very dear friend and part of the ImmuFar section that Francesca has developed in IUFAR together, okay? So Professor Carlo Riccardi is the director of the Department of Medicine and is a full professor of pharmacology at the Perugia University Medical School in Italy. He was the former president of the Italian Society of Pharmacology, and he will be sharing the role of glucocorticoids on immune regulation. Thank you, Carlo.
Thank you, Katerina. Thanks a lot. I would like to thank you, my chairmen, and Francesca that gave the start for the organization for the British Society of Pharmacology to this workshop. Actually, glucocorticoids are endogenous molecules, and they do interact with maybe many receptors, but we have to say it with glucocorticoid receptors. The glucocorticoid receptors are in all cells and interact with DNA because one of the main mechanisms is the regulation of gene transcription. And the recognition sequences on DNA as well as the receptor itself is in all cells and tissues. So when we give glucocorticoids, we should expect some effect everywhere in the body. All the chromosomes express many GRE in their composition. Just to say that it's a product of stress, one of the main response to stress, which make some problems. I mean, more studies are needed to understand well the relationship between stress and inflammation, stress and induction of glucocorticoids, and possible effect on inflammatory processes. It's not so clear, not so easy. Consequent to the presence of receptors in all tissues and organs, the therapeutic application of those molecules, or molecules derived from the structure of the endogenous glucocorticoids, are involved in therapeutic approaches to many diseases. Although maybe we do not cure really any disease. The possible interaction with immune response, let's say immune inflammatory response, which is more or less the same thing, is important. Like for example in the IBDs, that in fact are called inflammatory bowel diseases, but are autoimmune diseases with a component of inflammation as usual. This is typical of glucocorticoids and to interact with such kind of cells and factors and reaction and T cell memory. The T cell are important because they are responsible for memory. So if you have an autoimmune disease or an autoimmune-based inflammatory disease, and the disease will relapse because there is the immunological memory, and this is in T lymphocytes. Now, one point is, endogenous glucocorticoids are released from the adrenal gland. It's true. Many studies since more than 100 years, I mean. But there is also production of glucocorticoids into the tissues. So other than the adrenal gland, various lymphoid organs, including T cells, thymus epithelial cells, bone marrow, spleen, many tissues that are directly involved in the production and regulation of immune inflammatory processes. In the intestinal mucosa, which could be really relevant for some specific diseases, in the lung, in the brain, and in the skin, and the other tissues. So I don't know how much is important the production in the local production, but the question arises, at least for me, if when we administer glucocorticoid drugs, what we want to do. Maybe we want to restore the tissue-specific concentration of locally produced glucocorticoids. There is an indication of changes in the local production of glucocorticoids, not that I don't know, but I see many problems even in that. Interaction with the immune system, any kind of cell, of the innate or adaptive immune response. T, B, macrophages, dendritic cells. Virtually, glucocorticoids interfere with every step of inflammatory immune response. Usually is a depressive response, so it's an immunosuppressive anti-inflammatory effect. Although even in a simple scheme like this, you can find some increase of some immune response, such as the case that we all know very well since many years of the Th2. Th2 is part of the immune response, is part of immune-mediated inflammatory response, and it is clear that glucocorticoids upregulate the Th2. So on one side, restricting between Th1 and Th2, which may not be completely correct, but just to understand, there is an inhibition of Th1 response, increase of Th2 response. Another important point, it seems from literature, I'm not expecter of such kind of experiments, is the migration of leukocytes migration into the site of autoimmunity and inflammation. And here too, I mean, I have some doubt because the inhibition of many processing, which is addition, infiltration into the tissue, and so on, this is regulated by transcriptional regulation of many addition and such kind of molecules responsible of this important process. And so my question is that there is an inflammatory infiltrate. I imagine that you overcome the disease when cells come back from the tissue and are not more infiltrating the tissue, giving the inflammatory immune response and disease. But if you have an inhibition of traffic of cells in one sense, you may be the same on the other one. So you make a treatment for a disease with glucocorticoids, and then I don't know if you can obtain really a resolution of the inflammatory process. From what I said until now, it seems that I'm against glucocorticoids. Isn't true. It's my half of my life at least, but there are problems. I would say something about variability and adverse effect. Those are not all the problems I see in glucocorticoids, but just 5. So variability, adverse effect are there. Differences between single glucocorticoid drugs. All that is empirical. No one can tell us why you use this molecule instead of this other molecule. Dose and timing of administration. This is impossible to give a rationale. Glucocorticoid resistance, tolerance, dependence, which is a big part. I mean, I mentioned the Crohn's disease at the beginning. I can say roughly that when you make treatment with glucocorticoids, at least the clinicians say I'm not a clinician, and you may have a response in 40% of the patients. What about the other 60% Crohn's disease? Maybe 50% with ulcerative colitis. What about the other 50%? Same for adverse effect. Many tissues, many kinds of ADR. Receptors are everywhere. So if it's not an effect that we consider therapeutic, you have some kind of adverse effect. I underline the neuropsychiatric effect that was also in the first slide, but was too early at that time, because this is very, very interesting. There is a paradigm. I'm not expert in neurons and psychiatric diseases, okay? But from literature, you have stress, inflammation. Inflammation is essential to generate psychiatric diseases. Clearly, there are epigenetic events, otherwise you don't get the disease, particularly if you speak about the stress of the infant and then the disease in the adult subject. And stress, immune inflammation response, brain changes, psychiatric diseases. And what about glucocorticoids there? We can say, well, those glucocorticoids are there to antagonize somehow the inflammation induced by stress, so this is very important. But actually, just one slide, just one. Another doubt. There are immune-stimulating pro-inflammatory effects of glucocorticoids. This is a specific point taken from literature, and that deal with toll-like receptors and the NLRP3 inflammasome protein. Those molecules, those receptors, even in T lymphocytes, and by direct effect, not mediated by accessory cells, dendritic cell, direct effect, activate T lymphocytes, activate the inflammasome. Glucocorticoids upregulates such kind of response element. If you show just this slide to someone coming from another planet, another world, and you say, what are those drugs? Oh, those are immunostimulating pro-inflammatory drugs. Okay, I close here because I'm not prepared to do more than this. But clearly, I think from what I said until now, we have a problem of variability. Before there was the question, the good question to the beautiful speech by Katerina relative to the histamine receptors and sex and gender and so on. For glucocorticoids, there are completely different responses between males and females. So there is a very big variability. This is the receptor, virtually in a receptosome, very big, because all those interactions has been demonstrated on the basis of molecular studies. And after the binding of glucocorticoids, they translate to the nucleus and bind to glucocorticoid recognition element as monomer, as dimer, or as tetramers. It's possible. This is not only the only interaction with transcription, because there also is there are not direct genomic that are represented here. I mean, the receptor can bind to the transcription factors. Clearly, they have a sort of co-inhibitory activity, or maybe some of those migrate to the nucleus, and let's say in the tetrameric, this is an hypothesis, okay? In the tetrameric structure, they bind to DNA and they regulate all the genes that can be regulated. But then there are many more nuclear receptors. So it is true, the homodimer restricts on the dimer now, so otherwise we are lost. But you may have heterodimers with mineralocorticoid receptor, PPAR, estrogen, estrogens receptors, androgen receptors, and other. Virtually, the dimerization, heterodimerization, not homodimerization, can be true for any nuclear receptor present in the cell. Now, if you have a different dimer, the recognition on DNA will be different, with different affinity, energy, and capability to regulate the transcriptional activity. So how can you predict when a drug enters the cell what could be the effect? It's difficult. And this is the major difficulty we have in such kind of drugs that are very important, life-saving in many conditions, but then we are not able to proceed again to do better in our therapeutic approaches. I said already this is the dimer and the recognition. Dimer bind the Gerais and have different transcription activity. So we have many dimers, many transcription factors, and that the glucocorticoid receptors create homo and heterodimers, so it's a very unpredictable the response. If you go here, I want to recall at least Liberty, Berman, Sadlowski, many others that they made even more, but I cannot remember everyone. And you see there are different alternative splicings of the receptor. So till now we spoke about GR. There are 8 methionines for each. So from this slide, we have 40 different receptors that can form 40 homodimers plus, I don't know how many heterodimers. Actually, from literature, the splice variants are 17. Each of these has 8 methionine start point of translation of the receptor. So you may have 136 homodimers, different homodimers. And the number of heterodimers I am not able to calculate. In addition to the interaction with the other nuclear receptors. All those binds DNA in all cells and regulate. Usually, we say are anti-inflammatory. I mean, the receptor is morphogenic. I think the ancestral receptor appears 750 million of years ago, which means something. Then we may have a mutant with loss of function. Then we have post-translational modification that changes the transcriptional activity. Monomers, dimers, we said already that. Various heterodimers, interaction with cytoplasm signaling molecules. We have seen something in the cartoon with the cell and the nucleus. We don't have time to stay too long, unfortunately. And then the binding of other ligands. I choose the progesterone for this slide. This is in a physiological condition. In the pregnant women has normally levels of progesterone able to bind the glucocorticoid receptors. So it's some kind of stable anti-inflammatory condition or pro-inflammatory, I don't know. And clearly, we have no time to speak about that, other than the transcription effects. There are the epigenetic effects. Those are important. Still more studies needed. How to come out from this kind of very difficult situation related to drugs that are so relevant and important at the clinical level? One possibility is to study the transcription products and see if the early transcription product can have an effect similar to that of glucocorticoids, and then try to transform those molecules in drugs acting in vivo and having the potential of clinical activity. I mentioned here some, the most known, and those molecules could become safer anti-inflammatory drugs with less variability and less adverse effect, simply because are not any one of those is not a system so complex and like the glucocorticoid receptor system. There is no comparison. We studied GILs or GILs, and GILs are okay, so chromosome X. It's not so relevant here, and we found that at least 2 alternative molecules. This is the first one. This is the second one. We just call long, which fantasy, and can exist as a monomer, but even as a dimer, and interact with many, not like glucocorticoids, but with some other cytoplasm or nuclear molecules, such as RAS, RAF1, and Jun, Phos, SMAD, mTOR, and so on. Consequent to those interactions, there may be the immunosuppressive anti-inflammatory activity. But could be that true? Let's go and see. T lymphocytes. An example of glucocorticoids immune interaction. Now, the major effect of the glucocorticoids on T lymphocytes is the inhibition of cell proliferation. This is essential. When the antigen interacts with the T cell receptor, there is the activation of T lymphocyte that start to proliferate in part. Induction or inhibition of cell death. You may kill the T lymphocytes or make it surviving. That makes a big difference. And again, the regulation of cell differentiation. We spoke before about Th1, Th2. There is a clear switch of the differentiation in favor of Th2. I mean, this is very much from literature. All those processes require gene transcription. Now, in the next few slides, I want to convince you or show to you, I mean, not convince, that GILS is a very good molecule acting as immunosuppressive anti-inflammatory molecule. This is the possible effect of GILS on T cell proliferation. Glucocorticoids do inhibit cell proliferation, T cell proliferation. You see here the inhibition, but if you make a silencing of GILS, you lose the anti-proliferative activity of dexamethasone in this experiment. Here is the number of cells. It's not surprising because GILS binds RAS. And it's so true that binds RAS that when you activate with PMA the cells, RAS, translocated to the membrane and together with GILS. So it's a pretty good dimer that can go together to the membrane. Apoptosis, same story. The apoptosis, glucocorticoids can induce T lymphocytes apoptosis, not only T lymphocytes. Maybe I have a slide on this. And clearly, GILS is important for the induction of apoptosis. If you eliminate GILS, not totally, but you lose some of the pro-apoptotic activity of glucocorticoids. Not all. Glucocorticoids have, that I know, have many different mechanisms of apoptosis induction. But if you eliminate GILS, you lose something significantly. And this is the cartoon on apoptosis. Glucocorticoids interact with the receptor, and then start the direct and indirect control of transcription. There is upregulation of GILS, and this is enough of help for the caspase activation pathway, which means apoptosis. I must say that there is in the literature, these are other mechanisms that go through the mitochondria, and but I have some difficulty with this because usually, at least in our hands, which doesn't mean anything, but in our hands, if we use inhibitors of protein or RNA synthesis, we inhibit apoptosis. So other than GILS is not this important. An important thing here is that those apoptotic mechanisms are transcription dependent. This is true, but there is the variability, which is the context. So you have the T cell receptor activation, and you get 82% of apoptosis. This is the probability iodide assay. It's an older slide I like so much. This is always with me, this slide. If you give dexamethasone, which by itself induces apoptosis, you don't see anything. So it is true that dexamethasone induces apoptosis, but can also counter apoptosis given by other activating mechanism. And this is due to gills, because in the presence of other stimuli, gills, instead of participating to the apoptosis, is able to protect from apoptosis. In this specific experiment, because it's able to inhibit the Fas ligand expression. T-cell apoptosis start when Fas ligand is expressed on membrane. And this is the concept. You see, here you have non-activated T lymphocytes, which are highly susceptible to apoptosis. Then you have activated cells, which are protected, and maybe the hyperactivated are not regain the susceptibility to apoptosis. Here you have immunosuppression. Here you have survival of cells, memory of disease for us, because we worry about such kind of diseases. Antigen stimulates T lymphocytes, proliferate, differentiate, big apoptosis, some survival, clonal expansion. Treatment with glucocorticoids, gills upregulation, no proliferation, no activation, but survival. And you keep allergic cells there. And those are responsible for the recurrence of the disease after time. You do not eliminate the autoreactive clone, so you do not eliminate the memory of the disease. TH1, TH2, simply, I want to say that gills is able, like glucocorticoids, to change the ratio between GATA3 and TBET that is responsible of the switch to TH2 cells respect to TH1. So even in terms of differentiation, gills mimic the effect of glucocorticoids. Without going into details, is not. And what about T regulatory cells? It is known since many years that dexamethasone is able in vivo to increase the number of T regulatory cells. It's another differentiation example, which is interesting because clearly T regulatory cells are immunosuppressive, anti-inflammatory, may contribute to the final effect of glucocorticoids. If you eliminate gills, you do not induce any Treg. Here seems to be absolute the effect. It seems that without gills, glucocorticoids cannot induce T regulatory cells. This is because gills binds to the SMAD complex, which is and make the complex capable to regulate the transcription and FOXP3 production. If there is FOXP3, you have T regulatory cells with suppressive activity. If you do not induce FOXP3, no T regulatory cells. This is absolute. And so the story is that gills mediate TGF, GC-TGF synergism in FOXP3 induction. This effect, well known before, is even this mediated by gills. And this is what has been said before. So increase of TH2 and Treg, this is an effect of glucocorticoids. Inhibition of TH1 and TH17, at least in part. And gills makes the same, increase of TH2 and Treg, and inhibition of TH1 and TH17. So even in terms of differentiation, it seems to be similar. Does have any therapeutic effect? okay, this is more difficult in the sense that those are models, experimental models, animal models. But in the animal models of colitis, the short one by the NBS, the 5 days colitis, or the IL-10 knockout model, which requires weeks and months to develop and to cure, there is, in any case, inhibition of the disease when you inject mice with the fusion protein of gills, which suggests that the molecule may be active even pharmacologically, giving all the effects we have seen, favoring T regulatory, inhibiting autoimmune and inflammatory response. To conclude, I would like to say that clearly, as you know, all you know, glucocorticoids are widely used and they are effective in inflammatory or inflammatory autoimmune or immune-mediated inflammatory diseases. The major problems, but not the only one, is variability of the effect and the adverse reactions. And as I have tried to show to you, studies on protein peptide-based drugs derived from the transcription product could be helpful to find new drugs and with less adverse reaction and the variability of the effects, because this is the problem we have with glucocorticoids.
Thank you very much, Carlo, for this excellent yet non-exhaustive presentation of glucocorticoids, which we all know are very important. So this is now open to discussion. Please.
Okay. Marco Martins, Oswaldo Foundation, Brazil. Thank you very much for this very interesting talk. My question is there any evidence that gills could replace glucocorticoids under conditions of resistance or insufficient insensibility to glucocorticoids?
I think this is really a good question, and I don't know about. I mean, we didn't make such kind of experiments. We didn't try. It seems to me a beautiful idea, honestly. I mean, we will consider that. I think it's very important because the other important point is resistance. Can you overcome, other than have less adverse reaction and less variability, can you overcome the resistance and the dependence too? I mean, but I selected one piece clearly.
Thank you. Hello. Yes. Thank you for your comprehensive lecture. Fascinating of how much we still don't know these life-saving drugs. And also, my question relates to steroid resistance. What is your opinion of the steroid resistance, where molecularly it can happen? And I also was curious to know if a gills-based drug may circumvent the resistance that some people have to start with. I mean, in asthma, you know, the field I research, you have a high percentage of steroid resistance that are not resistant to the hormonal effect of steroids, but are resistant to the anti-inflammatory one. So what is your opinion? What can you comment?
My opinion and expectation is that could be very important. I think you made the point really relevant. There are not indications until now. I can tell you that in a few weeks we should submit a paper with many different peptides derived from gills. But unfortunately, the pathological model we are going to use is always mouse colitis. So, asthma would be really relevant too, as well as other kind of diseases. Let's see.
And a comment also, I don't know, what do you think, a comment and a question, what do you think about a paper that appeared during COVID by the group of Charles Sirhan, showing us that steroid might induce pro-resolving lipid mediators, such as lipoxin, resolvins, so might have also pro-resolving anti-inflammatory activity. I think from your lecture it can be.
I don't think that with glucocorticoids you can for sure obtain the resolution of inflammatory. It's too complex. Clearly there should be conditions in which you do, but you cannot predict, and you will have always a certain percentage of people responding to the attempt to have a resolution, otherwise you don't. The migration of leukocytes into and out the tissues is a typical example.
Thank you very much.
Thanks to you, Francesca, once again.
May I ask a question? I don't see anyone else. What about these peptides? Would they induce hypoxia in tissues, or this part of glucocorticoids, which is very important clinically, as we all know?
I don't know, but I do not expect. No, the peptide we made are in the proline-rich region of gills. I mean, you never know if you are staying time when you make presentation, you know. And but when I show the molecule as monomer and dimer, there is a big part of proline-rich region, and there around the residue 121, 122, there is a specific binding place, binding to one of the NFKB chains. So you are in the anti-inflammatory, theoretically, you are in the anti-inflammatory business. So the peptides are projected for this. But clearly one can start with the entire molecule and see if there is any effect. Then you can check for the specific region and construct the specific peptides. You have seen that in one of the figures I put RAS on one side of the molecules, RAF in another one. It's true, I mean, we made the mutation of the protein showing we lost the binding to RAS in one case, to RAF in another case, to both in another case, continue to bind when this is out from the proline-rich region, okay? RAS-RAF binds it on the other side of the molecule. It's a very complex, it's a small 120, 130 amino acid molecule, but already there is an entire world inside this story, this molecule.
Yes, that's a very complicated story, I understand, as usual with glucocorticoids.
Like glucocorticoids.
Yeah, yeah. Thank you very much, Carlo, for this excellent presentation.
So we're just checking one last time if Dr. Mayuri Tarasuk is present for her oral communication talk.
No?
No.
So I think we will skip then, since the lady is not here, and I would like to invite my co-chair, Hong Yong Peh. He is an instructor in Harvard Medical School in the US and an investigator in Brigham and Women's Hospital. He serves as the Young Investigator Chair in the Immunopharmacology section or committee these days of IUFAR, and his talk today will be on Resolvin D2 stimulates resolution and regulates type 2 responses in allergic lung inflammation.
Thank you, Katerina. Yep, so today I'll be presenting my work on Resolvin D2 in allergic lung inflammation. I would like to thank British Pharmacological Society for sponsoring this session, and as well as the IMUFA for putting up this proposal. So I'm sure asthma is a well-known disease to everyone, so I'll just give a very brief introduction. So the pathobiologic features of asthma include airway inflammation, mucous cell metaplasia, and airway hyperresponsiveness. Common clinical allergens include house dust mites. And for allergic asthma, the main inflammatory cells involved will be eosinophils and type 2 cytokines such as IL-4, 5, and 13. So some of the pathobiologic features of allergen-associated asthma can be modeled in murine allergic airway responses. So now I'm just going to give a brief introduction into resolution of inflammation as a follow-on from the resolution pharmacology session on Tuesday. So at the site of injury or infection, it will release chemical mediators such as cytokines, chemokines, and eicosanoids like leukotrienes and prostaglandins, and that mounts an acute inflammatory response. So this is meant to be a protective innate function of the body. But if inflammation is left unchecked for a prolonged period, it results in chronic inflammation, as you can see in rheumatoid arthritis. It also affects wound healing and organ fibrosis, as seen in diabetes, cardiovascular diseases, and asthma. So if we manage to contain inflammation at early enough, this process is known as resolution. So it was once assumed to be a passive process, but of course, now through the concerted efforts of our group and collaborators, we now know that resolution of inflammation is an active process orchestrated by specialized pro-resolving mediators, or we call it SPMs in short, to resolve the acute inflammatory responses. So SPMs are found at bioactive concentrations in human tissues, as illustrated here. It is present in all key aspects of the body, as well as it was also detected in breast milk, so further suggesting its biological importance. So SPMs are not only produced by mammals, but it's also conserved in marine life forms as well. So going back into the context of how SPMs and in the asthma, so for asthma, the initiation is usually by the allergen on the epithelial cells, which induce the shift towards a type 2 inflammation by T cells, and that recruits eosinophils and degranulation of eosinophils in the airways, goblet cells producing mucus, and accumulation of all these results in bronchoconstriction. So there are now some studies that we have published on the various SPMs on how we was able to phagocytose the allergens. It will reduce the leukocyte endothelium interactions, inhibits the leukocyte transmigration, clearance of edema, as well as inhibition of goblet cell hyperplasia. So in this slide, I will give just a brief overview of SPMs and how they are derived. So from arachidonic acid, we were able to get the lipoxins family. From omega-3 fatty acid, EPA, we were able to get the Resolvin E series family. From DHA, we will get the Resolvin family D series, as well as the protectin families and the myrisins families. So in my talk, I'll be focused on Resolvin D2 in preclinical asthma. So the biosynthesis pathway of Resolvin D2. So it begins from DHA, and in the presence of 15 LOX, it forms a 17S hydroperoxy DHA, and together with 5 LOX, it forms the intermediate of 7S, 8S epoxy resolvin, and in the presence of hydrolase, we get Resolvin D2. So Resolvin D2 now has been shown to block neutrophils infiltration. It increases a process known as efferocytosis. So efferocytosis is a process where macrophages phagocytose apoptotic neutrophils, and Resolvin D2 has been shown to decrease pain. So 3 selected publications from our group that we have shown that how Resolvin D2 is able to is effective in controlling sepsis, as well as improving post-ischemic revascularization, and Resolvin D2 in attenuating bacterial pneumonia. So there's this paper by Vickery and Tim that have shown that this regulation of resolvin D2 is affected in chronic rhinosinusitis. This paper as well showed that the DRV2, which is the receptor for resolvin D2, is one of the potential causal genes for asthma by the UK Biobank study. So going to the mouse model, in order to mimic the allergic asthma, we use the house dust mite as allergen in the mouse model. We give it once weekly through the intratracheal route on days 0, 7, and 14, and then we get the lung samples on days 17 and 21. So this is the targeted LC-MS-MS lipidomics against some of the icosanoids and SPMs. So as you see over here for the icosanoids like leukotriene B4 and the prostaglandins, they are actually increased on day 17 of the model and further increased on day 21. So conversely for SPMs like resolvin D series or mericins, they actually decrease on day 17, but you see an increase of their levels on day 21. So you can see like the balance between the icosanoids and SPMs. So of course, it's of interest to us that resolvin D2 was likely to be below the detection limit of the instrument or perhaps absent in the airways. So with all of this background that I've shown so far, our question is, what is the role of resolvin D2 in allergic lung inflammation? So going back into the same mouse model where we took the, so this time instead of taking the lungs, we actually performed bronchoalveolar lavage, so BAL in short. We took the BAL from mice on day 17 and day 21. You can see with house dust mite challenge, there's an increase of total leukocytes in the BAL, and there's somewhat of a partial resolution on day 21, but it's still inflamed when you compare it to saline challenge group. So being an allergic lung inflammation, the main inflammatory cell involved would be the eosinophils. As you can see on day 17, it accounts for 2/3 of the total leukocytes in the BAL. Of course, being an allergic model, there's lymphocytes that's involved as well. There's a very partial increase of neutrophils, but in this model, neutrophils are not really present or of interest in here. So now, with that overview of the asthma model that we have developed, so we try to get a therapeutic approach where we only introduce the resolvins after the final house dust mite challenge. So we give it twice, once daily on day 15 and day 16 through the intranasal route at a dose of just 100 nanograms. And then we will collect the samples on day 17, 19, and 21. So the total cell counts, as you can see, by resolvin D2, right from day 17, we was able to attenuate the total leukocytes in the BAL. And then, of course, which is of interest to us, is it was able to reduce the eosinophil counts right from day 17 as well. So as part of our group, we like resolution indices. So in order to better encapsulate the resolution of eosinophils, we have this resolution interval graph and table. So we define the resolution interval for eosinophils as a time taken to reduce the maximum eosinophil counts by half at Tmax, from Tmax. So you can see in Vigo group, it took over 3 days for it to reduce for the resolution interval. And when you give resolvin D2, it just took about a day to reach half of the eosinophil counts. So with this, we can see that resolvin D2 definitely accelerate the resolution of eosinophils. So again, being an allergic model, we do see the presence of lymphocytes, and resolvin D2 was able to attenuate the counts of lymphocytes. So now moving into the lungs, so we perform H&E staining. As you can see on day 17, there's a huge amount of cell infiltration into the airways, as well as bronchial epithelium thickening. And when you give resolvin D2, you definitely see a reverse of this. There's a less cell infiltration, and the bronchial epithelium is not as thick as the vehicle control. We next perform PAS staining. So it's just to stain for mucus. So mucus will be stained purple. As you can see in vehicle control, there's a lot of mucus cell metaplasia on the bronchi epithelium. And when you give Resolvin D2, you see the reversal of the mucus production. So now moving into a lung physiological measurement. So we use methacholine, which is a known bronchoconstrictor, and to measure the airway resistance of the respiratory system using the FlexiVent machine. So on day 17, as you can see, the in vehicle control, the mice airways are hyper-responsive, where there's a huge increase of resistance to the increasing dose of methacholine. And when mice were treated with Resolvin D2, you see that there's a reduction of the airway resistance. So on day 21, even though there was a reduced leukocytes in the BAL for the vehicle control, you see that the airways actually remain hyper-responsive, where the resistance data is still similar to that of day 17. So reduced leukocytes in the BAL, but you still see a hyper-responsive airways or resistance in this case. And when you give Resolvin D2, we promoted the resolution, and it brought it back to levels of naive animals. So in order to encapsulate the resistance better, we calculated the ED200 of the resistance. As you can see on vehicle control on day 17, there's a 4-fold decrease of ED200, and there's a partial increase by Resolvin D2. On day 21, vehicle control still remains the same as day 17, but you can see that in Resolvin D2, it was able to restore back to naive mice levels. So once again, a resolution indices from our group. So in this case, we define a resolution interval here for the resistance ED200 as a time taken to double the methacholine ED200 from T minimum. So in this aspect, a higher ED200 is actually good because it means it requires a higher amount of methacholine to induce the same amount of bronchoconstriction. So you can see in vehicle control, it took about 7 days for the resolution interval, and in mice administered with Resolvin D2, it just took 2.5 days. So again, we can see that Resolvin D2 promotes the resolution of methacholine-induced airway hyperresponsiveness. So going into some biochemical profiling, the cytokines in the BAL, we measured type 2 cytokines such as IL-4, 5, and 13. They are increased when challenged with house dust mite and reduced when treated with Resolvin D2. So again, Resolvin D2 promoted the resolution. As you can see by day 21, it actually brought the levels of the type 2 cytokines back to saline challenged animals. So IgE is also observed to be increased in patients with asthma. So we measured the total IgE levels in the serum. As you can see, there's increase of total IgE when mice were challenged with house dust mite, and this was reversed in the presence of Resolvin D2. So with all of the data that we have shown so far, so we now ask the next question. So are these actions of Resolvin D2 mediated by its receptor? So but before I dive into the receptor, I would just like to bring your attention to how we authenticate and validate SPMs in our group now. So starting from the top right, in the anti-clockwise motion, we will have the structure of Resolvin D2 and its proposed MS-MS fragmentation. In top left, the UV spectrum of Resolvin D2 showing a maximum absorbance at 301 nanometers. Of course, that's the lambda max in methanol. In bottom left, the retention time of Resolvin D2, and in bottom right, the mass spec spectrum of Resolvin D2. So Resolvin D2 has been published and known that it actually binds to DRV2, or also known as GPR18. So we measured the DRV2 expression on CD45 positive cells in the BAL for the animals. In gray, over here is DRV2 FMO. In blue, saline, and then in green, mice challenged with house dust mite. So you can see that in the challenge of house dust mite, we see an increase of DRV2 expression on the macrophages. And these are the relative expression of DRV2 on the various leukocytes in the BAL. So from this, it seems like macrophages and eosinophils have the highest relative expression of DRV2 in a BAL. Overall, when you give Resolvin D2, besides neutrophils, there is really no changes in the receptor expression. So it's suggesting that Resolvin D2 doesn't actually act by reducing the receptor levels in the bowel leukocytes. So in order to investigate if we have similar findings in patients with asthma, we managed to obtain human whole blood from the Severe Asthma Research Program, the Boston site. So we took whole blood from patients with asthma, and we looked at the eosinophils and compared against healthy volunteers. So as you can see, in patients with asthma, there are increased eosinophil counts in the blood. And of course, they do have increased DRV2 expressions as well on the eosinophils. So we see similar findings in both human with asthma, patients with asthma, as well as in our asthma model. So now, going back into the mechanism to see if the effects of Resolvin D2 will be affected in the presence of the DRV2 antagonists. So O1918 is the antagonist. We introduced into the mouse model 30 minutes before each Resolvin D2 dose, and we give O1918 via intraperitoneal injections. As you can see over here, the dose of the antagonist is almost 250 folds higher than what we give to Resolvin D2. So back into the BAL leukocyte counts, we can see that Resolvin D2 was able to reduce the leukocytes in the BAL, and this effect was reversed in the presence of O1918. So something that was interesting to us, particularly the macrophages. So macrophages, of course, we did by flow staining, and they are CD68 and F480 positive, and you can be split into three what we call subpopulations: infiltrating macrophages, exudative macrophages, and alveolar macrophages. Sorry for the typo. This should read as CD11B. So yeah, CD11B and CD11C. So interestingly, Resolvin D2 actually seems to only reduce the counts of the Imax and the Xmax, but it has no changes on the alveolar max. So suggesting that there must be some form of specificity for the Resolvin D2 to only affect the activated macrophages. So moving from the BAL into the lungs, we measured the counts of the CD4 T cells and ILC2s in the lungs. There's a partial decrease by Resolvin D2 on CD4 T cells, but there's a significant decrease on ILC2s. And it was reversed in the presence of O1918. So breaking down the CD4 T cells into the IL-5, IL-13, or both expressing receptors on the surface, we see that there's increase of this type 2 cytokines receptors on the surface when challenged with house dust mite, and then it's reduced when given with Resolvin D2, but this was increased again when in the presence of O1918. We observed a similar data on ILC2s as well, on the IL-5, IL-13, or both expressing receptors on the surface, where Resolvin D2 effects were reversed in the presence of O1918. So lastly, just to summarize what we have shown so far, we have presented work in progress for this where we administered Resolvin D2 into this asthma model. Resolvin D2 accelerated the resolution of the BAL inflammatory cells, particularly by shortening the resolution interval of eosinophils. It reduced type 2 cytokines, mucous cell metaplasia. Resolvin D2 was able to resolve the airway hyperresponsiveness. There are increased expression of DRV2 in house dust mite challenge animals, as well as in patients with severe asthma. And in the animal model, we see that these receptor-mediated actions by Resolvin D2 were reversed in the presence of its competitive antagonists. So to conclude, Resolvin D2 decreases type 2 inflammation and promotes resolution of allergic airway responses, thus suggesting that the Resolvin D2 DRV2 receptor axis could be a potential target for disease modification in asthma. Lastly, my acknowledgements to my mentor for 6 years and counting, Dr. Bruce Levy and his Air Lab. All of the co-authors of the manuscript are actually highlighted in bold. So Dr. Levy will be giving his talk tomorrow at 9:45 a.m. I also like to thank my co-mentor, Dr. Charles Serhan, as well as the fellowship and the various fundings. Lastly, the severe asthma research program as well at Boston. Thank you.
Thank you very much. Is there any questions? You can queue in the...
Yes.
Okay. Mauro, yeah.
It was a very nice presentation.
Thank you.
And I don't know what I'm talking about because I do essentially neuropharmacology. No. The stupid question of the day is, in your asthma model, what happens on day one? And I will explain why I ask what happens on day one. I'm in Melbourne, and sometime in November 2015 or so, there was a massive big potato in the middle of the country, and that moved then fast forward. And we experienced something which was called a thunderstorm asthma. Within about 18 hours, we had 9,000 to 10,000 people coming to emergency rooms with massive asthma attacks. 9 of these, or 10 of these people died. This was a very rapid effect. I think that half of the population had never had asthma in their life before. Yet these people had very severe clinical findings. So question again, what happens in your model on day one, not on day 17 or 21? Have you ever looked at it?
Yes, I did. We definitely look at it. But I guess in this context, to answer your question, there's some background is, in these animals, they were not exposed to house dust mite at all throughout their lifetime. So on day one, when you give house dust mite, the first response is a neutrophilic infiltration. So you see a huge increase of neutrophils in the airways. They do not have a precursor response of the type of the T cells against this house dust mite. So it takes about 7 days for the T cells to be primed towards a type 2 inflammation, and then that results in the recruitment of eosinophils. So eosinophils comes in about day 8 of the model. But in terms of some of these thunderstorm asthma that you see, I'm not a clinician, but in my opinion, I think that these patients could already have some pre-exposure to house dust mite or to whatever allergen that caused the asthma. So the moment they are already primed, so the moment they have it, there's perhaps a huge inflammation in these patients, and it gets overwhelmed. I'm just wondering, did you actually measure in these patients if they have like higher levels of eosinophils or neutrophils?
Professor Alistair Stewart is going to ask this question, or answer it.
Thanks, Danny. It was November 16, 2016, a real catastrophe for Melbourne and the surrounds. I'm interested in the connection that you make between Resolvin D2 and resolution of infection, because the model that you've chosen to use is a very steroid-sensitive model, whereas if you complicated the model with a viral or a bacterial infection, then it would become less steroid-sensitive. It seems to me that the pharmacology of Resolvin D2 fits with a model where there is complication by viral and bacterial infection, and I'm now asking what you've looked at in that space.
You're right. So we haven't done a model where we give house dust mite with a secondary virus, or at least I haven't done it yet. That's something we could look into. But with that said, Dr. Levy's lab, the Air Lab, have done a lot on resolvins in virus work. I think there was a paper published in MBio where they did a 2-hit model. So it was a bacterial and then followed by a virus model. You can see not resolvins, but in that paper, it was mericins. It was able to protect the animals when they have a 2-hit of a bacteria and a virus. So yeah, that'd be interesting to take a look if Resolvin D2 will work in a more glucocorticoid resistant model.
Yeah, thank you. That's where the unmet need is.
Yes, definitely.
Hi, I knew that you will have a great presentation.
Thank you.
We know each other from Dr. Bruce Levy lab. I wonder about the specificity of the antagonist you used, that is one available, Since it is also acting on GPR55, that is a supposed receptor for endocannabinoids and also synthetic ones. So do you think, based on that, it's just a speculation, that the specialized pro-resolving lipid mediators might interact with another pro-resolving anti-inflammatory endogenous system that has the endocannabinoids?
So
it's more speculation.
The quick answer would be, I believe it's yes. We think SPMs are really wonderful, and they really drive the resolution of inflammation in many aspects, not just in the airways, but really in multiple modalities of the body. I don't know the literature well enough if we have looked at basal sites, but I think it's most likely the answer is yes. Okay. So in particular for like Resolvin D2, it's actually shown to be produced in endothelial cells, neutrophils, and macrophages. So different parts of the SPMs are actually shown to be produced by many different cell types in our body now. So I think it's yes.
Thank you.
Hi, Hong. Chengxue here. Thank you so much. Congratulations on the comprehensive work, and it's really exciting to see how Resolvin D2 in this space.
Thank you.
My question is, so we heard quite a lot of resolution pharmacology and SPMs in the last few days. I wonder, do we know what's happening after the receptor gets activated for Resolvin D2? And if there is a convergent pathway for all the SPMs and those pro-resolving GPCRs intracellularly?
Thank you. In this case for GPR18 or DRV2, yes, there are some studies that have shown that when there's a ligand that binds to the receptor, it actually activates the phosphorylation of the ERK and certain of the pathways, so that's where it drives the transcription. In terms of do all SPMs converge into a similar pathway, I think the very broad answer is not really, because different SPMs actually binds to different receptors. So like for my case, Resolvin D2 binds to GPR18. We have the lipoxins that bind to ALX FBR2. And then we have Resolvin E1 that binds to the CHEM R23. So I guess that's actually while they are similar ultimately in functions, but I think there's definitely some difference at the transcriptional level.
Thank you. Really enjoy your presentation work. Thank you.
Thank you.
Hi.
Hi.
Hello. Thank you very much. Very nice data. I wonder if you have got any data on allergen-induced lung tissue remodeling in your model. I don't think I have seen any data on things like collagen deposition.
No, this is a somewhat acute model. It was done within 3 or 4 weeks per se. But no, we haven't looked at a much longer chronic model to see whether there's like changes in the smooth muscles and collagen. That would be something that we will definitely look at in our future works.
And a question from me.
Yeah, sure.
What happens if you give aspirin to the Resolvin production and induction?
So when you give aspirin, there's definitely a aspirin triggered so aspirin triggered usually creates the R stereoisomers. So we can have aspirin triggered Resolvin D1 or lipoxins. So so when you give Resolvins, they definitely have a production of the SPM as well. But in terms of giving aspirin to induce the production of the SPMs in the model, I think the short answer is it's not going to work very well, because you're going to really rely on the mouse cells to produce a large amount of Resolvins, or in this case, Resolvin D2. As you can see, Resolvin D2 was almost absent in the lungs. And we give exogenous dose of 100, even though it's very low, but it's 100 nanograms. It's going to take a lot of, it's a huge burden on the cells to produce such a large amount of Resolvin D2.
But is there any model to test the aspirin induction of resolvins like there is in the
Oh
eye?
yes, definitely. So yeah,
I mean, in the lungs, would you suggest something like that? Not on the eye. I know the work in the eye.
So
Because aspirin-induced model asthma is very important, particularly. Yes. So would you suggest any model to use or no?
So I do not know the literature fully well. I mean, perhaps Dr. Libby will have a much better answer to that question. Sorry.
Yeah, this was part of my worry about the asthma models and how we can have some firm conclusions on what happens on the pathophysiology. So that would be an interesting discussion.
Yep.
Yeah. Yeah.
So, very quick question. Rob Newton, University of Calgary. You may have said it, I may have missed it, but could you comment on the coupling of these pro-resolving receptors? So which GPCR pathways are they, which G protein pathways are they going down?
So different SPMs will bind to different receptors. We haven't mapped like all of the SPMs to various G protein coupled receptors, but in this case...
Do you have a feel for which G proteins we're actually talking about? So GS, GQ, G1213, GIO, what are the main pathways that you perceive as acting to do these things?
We know Resolvin D2 binds to GPR18, but in terms of the full, like the subunits of the G protein coupled receptors, that's something that I personally haven't looked at yet.
Right, thank you very much for this very interesting talk.
Thank you. So while I'm still here, so I'll introduce our final speaker for the day. Professor Mauro Teixeira is at the Federal University of Minas Gerais, and his group is the leading inflammation research center in Brazil, and he focuses on the inflammatory responses, infection, and inflammation. He'll be sharing the role of pro-resolving molecules and their potential clinical applications in infection and chronic inflammation.
Thank you. So thank you, everyone. Thank you, Francesca, for the kind invitation. It's a pleasure to be here. How do I start? Okay, good. So, not working? Yeah, it's working. I have to point here. Okay. So I will try to discuss with 3 topics. The first one was really helped by the previous speaker, so thank you, Dr. Peh. We'll briefly discuss resolution of inflammation and then discuss the potential role of that as a treatment in chronic disease other than asthma, and then touch on the role of applying resolution pharmacology to infectious disease. We see inflammation we want to treat because it hurts, it decreases life expectancy, and in the case of infections associated with tissue damage. And pretty much what we have done in the previous years is really to prevent the arrival of cells or prevent their activation once in tissue. Now, the concept of resolution starts with the production of pro-resolving molecules in tissue. So we don't really understand the switch between anti-inflammatory and pro-resolving molecules. The SPMs provide one of the possibilities, but there are, at some stage at the peak of inflammation, you actually have the signals which drive the pro-resolving molecules. And once these molecules are produced, there are a series of events that are really crucial to clean the tissue from. So there is death of the cells by apoptosis. These cells are eaten by macrophages, a process that we call efferocytosis. You stop the arrival of neutrophils, you make them go back, non-inflammatory monocyte recruitment, and as these cells come in, they eat dead cells, they reprogram in such way that tissue tends to return to the normal hemostatic levels. So these are the crucial processes that we define a mediator as a pro-resolving molecules. One mediator should induce granulocyte apoptosis, it should induce efferocytosis, it should induce non-inflammatory monocyte recruitment, and macrophage reprogramming. And clearly, this is not a passive process. You do need the production of mediators of inflammation, and we name these mediators resolution of pro-resolving mediators. So this is just to make the point. I mean, there is much more than the lipid mediators. These were the first ones to be found, but clearly there are protein, peptide, gases, short-chain fatty acid, hydrogen peroxide. These are all molecules which have been clearly shown to induce resolution of inflammation. And the point that was asked about the G protein coupled receptors and downstream signaling events, you know, there's an important role of cyclic AMP for some of the receptors, but for most of them, we simply don't know the signaling. I mean, this is a big area that's missing in the field, you know, the actual transduction pathways downstream of the molecules. So the concept here is that because we have this process that actively resolves inflammation, if that process is wrong and it's not occurring, that may actually contribute a lot to chronic inflammation, right? And so if you have a process that contributes to chronic inflammation, this is just the drawing of that idea. So in usual terms, you have resolution of inflammation that occurs in most circumstances. If this goes wrong, especially in chronic inflammatory disease, that may drive the inflammatory process. We're not saying that, you know, arrival of a lot of cells don't contribute to chronic inflammation, but their persistence may actually be a key driver of chronic inflammation. And that may be because there's a loss of that capacity to resolve. And the concept, the pharmacological concept behind this now is that if you add the pro-resolving molecule, you will eventually be able to make the tissue go back to normal, and this is what Mauro Peretti has dubbed resolution pharmacology. So this is what we are seeking from the point of view of applying resolution of inflammation to chronic inflammation. I will talk about one of these molecules, especially angiotensin 127, as an example of the possibility of this of these molecules in the context of acute inflammation and infection. So angiotensin 127 is a metabolic product of angiotensin 2 after it is metabolized by angiotensin converting enzyme 2. So it's a 7 amino acid peptide, and it binds to the MAS receptor. And by doing that, and we have no idea of the transduction events downstream of the receptors, G-alpha I, it will actually have a lot of anti-inflammatory events, or in general terms, it counteracts the effects of angiotensin 2 on the AT1 receptor. So these are receptors that almost a lot of the times have opposite effects. We have shown in the past that if you give angiotensin 127 or molecules that mimic the action of angiotensin 127 on the MAS receptor, you prevent the occurrence of inflammation. And we've also shown that if you give angiotensin 127 after inflammation has occurred, and this is the concept behind resolution. It is about treatment after things have occurred and not giving it before to prevent inflammation to occur. So clearly, angiotensin 127 decreases inflammation in models of arthritis, neutrophils in arthritis, eosinophils in acute and more chronic asthma. Now, so basically the concept is here, you immunize the animal, you challenge, and at different times after the challenge, you give your mediator and see what happens. You see the decline of inflammation. So there will be less neutrophils in comparison to control, but they were there already. So you're making them go away, and this is, in general, a process associated with an increase in apoptosis. And if you give an apoptosis inhibitor, you prevent that from occurring. You see that in human cells, so human neutrophils, angiotensin 127 will kill neutrophils or eosinophils by inducing apoptosis on them, and this is MAS receptor dependent. So I showed you two things. It kills and it makes the neutrophils go away from tissue. More recently, we have seen that, for example, angiotensin 127 will induce the migration of monocytes to tissue, and that migration is similar to a very well-known chemokine, CCL2. so it will induce their chemotaxis of macrophages, raw cells, cell line, BMDMs, or in vivo. And the effect is in general very similar to CCL2. If you inject angiotensin-127, it will actually induce CCL2. CCL2 is important for the recruitment, so it induces CCL2, and it cooperates with the receptor to induce recruitment. And the cells that arrive are actually tend to produce IL-10. Indeed, if you look closer, there is no change in the M1 phenotype. So, and as was also seen before, once you give angiotensin-127 to the macrophage, you tend to polarize them into M2 MRES phenotype. And because they have this phenotype, they are capable of phagocytosing dead cells or bacteria and killing bacteria. So clearly, angiotensin-127 is a pro-resolving molecule. It induces efferocytosis, it induces apoptosis, induces arrival of non-phlogistic monocytes, and when these monocytes are there, they are polarized to M2 MRES phenotype. Now, and I've also shown you that angiotensin-127 has clear apoptotic and pro-resolving effects in models of acute and chronic inflammation. Now, is there a role of this pathway in infection? And this is behind a whole line of research in my group that we are interested in trying to develop anti-inflammatory drugs to treat infection. Obviously COVID brought this to the spotlight, but this is something that people have been trying for a long time. And the idea being that, when you have an infectious disease, that there is a lot of tissue damage, in addition to the antimicrobial or if it's bacteria, antibacterial compound, antiviral, you will add something on top of that that will prevent tissue damage and will prevent the excessive leukocyte migration. So you need the leukocytes to be there to have an immune response, protective, adaptive immune response as well. But if it's too much, there will be tissue damage, and that's what you want to try to avoid. And so the idea is here, can we use the concept of resolution pharmacology and apply it to infection and see what happens? So I will use a model of influenza. I don't need to tell you that influenza is very relevant. It used to be the most relevant lung infection before COVID. And it will become again as we get good vaccines against COVID. And I will use a angiotensin-127 formulated in cyclodextrin. I don't have time to show any of that, but as you do that, you make this peptide amenable to be given via the oral route, right? So it is orally active angiotensin-127. I'll show you 3 models in where you have severe influenza. So you give a high inoculum. So if you give a high inoculum, influenza will kill most of the animals, about 50%. You can actually do that with most infections. You can give a lot or less. It will also show you a model of streptococcal pneumonia. And again, here you can give a lot of the bacteria or not so much. When you give a lot, there's a lot more pulmonary damage. Now, there's an interesting possibility as well. You can give a low inoculum of influenza, which will, there will be some suffering to the animals, but they all recover. If you go ahead and go 14 or 21 days and now give a very small dose of streptococcus that on its own does very little, that will actually kill the animals. So this is really interesting because that's what happens in the context of influenza infection, is that you may become infected by bacterial infection, and then it becomes much more severe. There are many reasons for that, no time to discuss that, but one of them is that the macrophages may go down, and as they go up, they are different macrophages, less able to deal with the bacterial infection. So these are the 3 models that we will discuss. And most of the time, we will be giving angiotensin-127 days after the infection. We are not giving preventive treatment. We are always giving therapeutic schedules. So we give a very high inoculum into the 4 PFUs of influenza. It will kill about 50% of the animals. Clearly, if you start treatment on day 3 and carry on treating, you will see that most animals will survive. They survive because there is less neutrophil infiltration in the lung. So the whole environment, and you can see, I'll go back, the tissue damage is decreased if you measure a score of inflammation. And interestingly, less tissue inflammation is associated with actually less viral replication. So, and presumably you have less signals for the virus to survive and replicate. So clearly giving angiotensin 1-7 after infection will prevent the severity of the infection. And we also see that one of the possibilities that angiotensin is doing is that it is killing the neutrophils by apoptosis, 2 different ways of doing so, and that neutrophils are being taken up by macrophages. So you have neutrophil apoptosis and their efferocytosis, and that's associated with an environment that decreases viral replication, and there is less pulmonary damage. Now, if we do experiments in the absence of the receptor, you will see that animals die much more, so disease is more severe in the absence of that break. And also, you can see here that if I give angiotensin 1-7 to mice that don't have the receptor, it doesn't work. So angiotensin 1-7 is protective in the course of influenza because it binds to the mast receptor, and that decreases inflammation. It induces resolution of inflammation, and you have an overall picture that is effective in preventing disease. Now, going to streptococcal infection, we can give a very high dose of streptococcus and start treating. Bacterial infection in mice, it's much more, it's much faster. So you don't have a very large therapeutic window here. So we're looking at 48 hours and start treatment at 12 and 36, just 2 treatments. And clearly, you see decreased neutrophil infiltration, but also decreased numbers of decreased damage as seen as protein in bowel as a marker of pulmonary damage. Very striking, you see a decreased number of bacteria. So there is for whatever reason, it is antibacterial. I don't have time to show you, but what we see is that angiotensin 1-7 activate macrophages to take up bacteria and kill bacteria, but very briefly. And also, you see less bacteria going to the blood. So there is about 50% lethality. So in some animals, there is a bacteremia, and angiotensin 1-7 treatment prevents that. It prevents bacteremia because it actually induces a range of proteins which are associated with the epithelial barrier. So there is an enhancement of the epithelial barrier. As you enhance the epithelial barrier, you prevent the bacteria from going from the bowel into the blood. So there is an overall protection, and this is associated with a much better damage to the lung. And I'll drive you through this. So I showed you that if we give the protein, so these are animals which are infected. These are animals which are given angiotensin 12 hours. If you delay angiotensin 1-7 to 60 hours, you lose protection entirely. Infection becomes too severe. Now, if we give antibiotic, again, starting very late, 60 hours, very small protection. If you now use antibiotic with the pro-resolving molecule, there is a very significant synergism and a lot of protection, right? So this is quite interesting because, there is a possibility that these pro-resolving molecules will be useful in the context of bacterial infection. This is something that we will try to look into. Now, to finalize, so I showed you that if you give a small inoculum of influenza, come along with streptococcus, an inoculum of streptococcus that doesn't kill 10 to the 3, it doesn't kill under normal circumstances, will actually kill 100% of the animals if you give after influenza. This is because the bacteria gets into the bloodstream. If you treat with angiotensin 1-7 just during the influenza, you prevent that late death, right? So basically, I've shown you 3 examples in which giving pro-resolving molecules is relevant in a model which mimics fairly reasonable what we see in humans with influenza. So the administration always favors the ability of the host because it has effects which are beneficial. Very briefly, this is not something to do with only this molecule. Other molecules will do similar effects. I'll show you some effects with this lipoxin analog produced by Catherine Godson in Dublin. So if we give this lipoxygenate 4 analog, and again, it's given after infection, and this is a model of severe infection, you will see that it tends not to have a major effect on the viral load, but it does decrease the number of neutrophils in the lung, and it decreases neutrophils because it increases their death by apoptosis and their uptake by macrophages. And by doing so, it will actually enhance the survival of animals after infection. So as a proof of concept, we do believe that enhancing pro-resolving pathways may prevent disease and either decrease infection or not alter infection, and this is actually quite good. So it may be that resolution pharmacology may be useful as an anti-disease strategy. Okay, and obviously it's not infection specific. It's not about it's not an antimicrobial, it's a disease, it's a strategy aimed at the host. So can we use resolution pharmacology in humans? And the question to the field is which are the molecules that are useful? So there are several that are in clinical trial. I borrowed this from Mauro Peretti. I will focus on this molecule. It's AP1189. We had a chance to lay our hands on it. It's currently in phase 2B trials for rheumatoid arthritis. So it is part of the proteins that are in the context of resolution inflammation. And AP1189 is a biased MC1, MCP, MC3 receptor agonist. It has anti-inflammatory and pro-resolving abilities. It has no melanin-inducing activity, so it doesn't activate the cyclic AMP pathway. It activates the GI pathway to induce resolution of inflammation. Okay, so using this molecule, we conducted an investigator-driven trial of the administration of AP1189 in patients with COVID needing respiratory support, not via intubation, but only needing oxygen. So it was a double-blind, placebo-controlled, randomized, investigator-driven trial. So the company gave us the compound. So, and we did it in Brazil in June 2021. It was mostly, what's called the gamma strain of COVID. So the design, we actually had 6 individuals in an open-label trial. We wanted to make sure that it was safe to give the drug to patients. At the time, the compound was given in a solution of 50 ml. If you've seen anyone with COVID in a hospital needing oxygen and needing to take 50 ml of a glue, it was a nightmare. So we would have to wait about 15 minutes to actually get, take oxygen, try drinking, put the mask in, breathe a little bit. It was a bit of a struggle. But anyway, so we managed to do that in the 60 individuals. And the primary endpoint is time to respiratory recovery. How long does it take for me to remove the oxygen from you? So how long does it take for me to remove oxygen and have a saturation of O2 greater than 93 for 30 minutes at room air? Okay, so we triaged about 400 patients and eventually had 60 individuals included in this study. Individuals were in their 50s. The body med BMI is not there. But they were, not many of them were obese, but they were all overweight. I think there was, age and weight were this, and age, sex, and weight were the most important thing that you could actually see at ICUs. They were moderately severe, as seen by this NIS2 score, and by the amount of oxygen they needed to keep a saturation above 93. Okay, so if we look at that, the time to respiratory recovery was very much decreased in these individuals, even if this is a small clinical sample. So most of the individuals on treatment blue actually stopped taking medication much earlier than individuals that took placebo in black. Because we removed oxygen from them earlier, they could go home faster. So on average, the mean time to hospital discharge was decreased in individuals taking the drug, the compound. And overall, there was a tendency to decrease, but the N was too small. Admittance to ICU, number of days of intubation, everything else. In terms of the safety aspects, there was much more nausea in individuals taking the compound, but there was nausea in almost everyone. It's just the severity. You know, taking a 50 ml of glue is not good. But there were no limiting side effects that would prevent the drug from being taken. So as a conclusion, you know, we added AP1189 to individuals already taking glucocorticoid. So that was the standard of therapy at the time. So individuals was mostly taking glucocorticoid. They were all taking, because that was the hospital policy, antibiotics at admission, and then it would be removed if deemed necessary. And most of them would be eventually taking heparin, and IV heparin if the D-dimer was high. So it significantly reduced time to respiratory recovery and discharge from hospital, reduced fraction of severe events, and no treatment side effects. So as a take-home message, you know, I would like to leave with you that resolution of inflammation is a very interesting new opportunity for therapy, and it has very good evidence now that it blocks aspects, unwanted aspects during infection. And I hope I've shown you and somehow convinced you that there is good evidence in humans that may be useful as well. So eventually we may be able to use resolution pharmacology as an anti-disease strategy. And I do hope in the future that most infectious disease, especially if deemed severe, will be actually treated with the antiviral or antimicrobial agent plus a drug that changes the course of disease. So I thank the resolution of group. The influenza studies were mostly done by Elisa with the help of my colleague Vivian. The clinical group was very large, so I thank Mauro and Thomas for the support with the clinical studies as well. And I thank you very much for your attention.
So we open the floor for questions. Yeah, please go ahead.
Hi, really nice talk. Thanks for taking me through Resolvins and the pharmacology. I guess my question's around, do you know much about the different systems of Resolvins? Do they synergize? Are they additive? How do they work together to resolve different diseases? For example, does infection have its own particular sensitivity to certain Resolvins? Chronic inflammation another set, or do we not know yet?
So the question is not resolved. I would say that, you know, annexin-A1 is quite central, it seems. So a lot of the pro-resolving molecules, lipids, melanocortin, will actually end up producing annexin-A1. Now, they do synergize with each other. So, but we, you know, I think it's a very fair question. We are trying to address it very, very much, and we certainly do not know the impact and the synergism between these molecules as yet. And a lot of it because we don't know a lot what's happening inside the cell. I think, you know, this is something that the field needs to sort out much better.
Mauro, that was a great presentation. Now, Ed Bullmore, on the first morning, made the point about the relationship between inflammation and depression, a number of other pharmacological, neuropsychiatric disorders. Now, is this applying to the brain? What do you know about this in the brain?
There is some evidence that it will protect acute damage, but that's pretty much what I know. I don't know if others could add to that.
And which are the target cells?
Because that is a problem.
Microglia? Is it?
Yeah. So target cells can be the leukocytes. A lot of the tissue, living cells, endothelial cells even, very much so. We've seen that in dengue. I would say the microglial cells and astrocytes would certainly be targets for these elements, but I haven't seen a lot of work studying these, especially in more chronic models where you have this sort of degeneration. In my mind, you have to remember these molecules, they make macrophages eat, right? Bacteria, dead cells, and when they make the macrophage eat things, that doesn't activate the macrophage, but it kills bacteria, but it doesn't activate them in terms of producing things. So I've always wanted to do Alzheimer's disease models where you need to eat things. Mm-hmm. They are there sitting around, and if you eat them, you die more. So, but it hasn't been resolved.
Yeah,
I've been trying.
In that case, it's only inflammation. It's not related to bacteria. And well, there was some point about long-term effects of viruses on, neuropsychiatric disorders, certainly in schizophrenia. And now we have long COVID where we may have something similar. But in most cases, people suffering from depression, there's no evidence that, no. But anyway, thank you very much. Great presentation.
Sorry, if I may just interject over here. So SPMs, I don't have the details of all of the diseases, but SPMs have been shown to be able to attenuate some neurological diseases. Yeah. Like Resolvin E1 is actually has been shown to suppress some form of depression.
Beautiful talk, Mauro. Congratulations. It was really amazing data. So I have two questions. One related to your first part on and 1 to 7. So it's during influenza infection, one of the greatest complications is secondary bacterial infection. And in part, the mechanisms that has been highlighted associated with that is increased levels of IL-10 that would reprogram macrophages, leading it to facilitate secondary infection. Differently, you showed that ANG1 to 7, although it induces IL-10, you showed this, that is controlling infection. How do you see this data? What are the mechanisms to control bacterial infection in this case? And my second question relates to your clinical trial, which again is beautiful data. And I'd like to ask if you are continues to follow up those patients. So as it's been clear now that long COVID is posed as an important public health problem, are you continuous to follow up those patients to ask if the treatment would decrease or would modify long COVID in this cohort?
So that is, no, we couldn't follow them up because there was the infrastructure disappeared. We were conducting a vaccine trial at the time, so it was way too much on the cup. As for the first part, it does do IL-10 as well, but it changes a lot the macrophage phenotype, a lot. So if you phenotype macrophages that are in the lung before and after these pro-resolving molecules, they are less inflamed. So when you do influenza, macrophages go down almost to very low levels at day 7, and then it starts going back up. Day 14 is almost normal, so that's why we choose that time point. On day 21, number of macrophages are down, but the macrophages are different. So as you recolonize, the pro-resolving molecules favor colonization with macrophages, which are MRES type and better able to deal with infection. And some of it has to do with the expression of PAF receptor on the macrophage, because the PAF receptor is one of the receptors for the bacteria to be eaten up. So, So there is a molecular mechanism, I just didn't have time. So there was some beautiful work from Luciana when she was at Bruce Lab that we actually show PAF receptors to be, at least for maresins, but also it seems for angiotensin 127.
Hi, Mauro. This is Helena from Melbourne. Really enjoy another talk from this conference. I was wondering your thoughts on the timing of administration of pro-resolving mediators. If you apply to resolution index, the nice time course that Hong showed previously, do we apply at the start or do we apply at the peak of turning on the resolution phase is more ideal? Thank you.
So we try to go for the peak, always. For the infection systems, this is hard. If you start delaying, you don't rescue, right? So in the clinic, and this is the hard thing about patient trials, is that, you can't delay administering, the compound, and there's a lot of heterogeneity in the patients coming in, so it's harder. But always the peak, and I suppose anyone trying to do it, and that was strong for COVID and dexamethasone as well. If you start giving it too early to too many people, you start seeing side effects that are not wanted. So I don't think the anti-inflammatory drugs for infection will be something that will be given early to everyone. It will be given with antibiotics, with everyone with signs of severity. Right?
Great. Could I follow up a small question? We have not enough time.
I'm afraid we have to end the session because
there's another one.
There is another. But I'm sure that you will have plenty of time to meet Mauro after the session and ask all these questions. I'm really sorry, and I thank very much everyone who participated in this very lively session. And of course, I would like to thank Francesca, who proposed the session, and the organizers who accepted this proposal. Thank you very much. Thank you.